Miss Sadia Momal.
Yes, you can see that.
Yeah, where is my screen?
उस पे आ रही है.
आ रही है? किस पे आ रही है? Okay, so what you can see in the screen, you can see R or you can see the R codes. क्या दिख रहा है आपको? Word file, and this is the file which everybody should have it, and that is where we can I can just copy and paste the codes from here. Have I did I take this R practice thing? ये हो गया था पीछे ये वाला सारा. I think we did that in metagenomics also. DG. So DG part is down there. It's here, in fact, from this line number 128. 128, and then you can scroll down. Everybody should scroll down. We started recording. Okay. And you copy this thing. You copy. And then once you copy, then you go to R. This is R, and let me make its graphics little bit bigger. So what I can do is GUI and 16 is, I think, okay. So that one is good, visible, right? So now next what we can do is I can I can write the code here. That's what we used to do in the previous sessions or classes. But today what we're gonna do is I think tomorrow I have a DB class. So, yeah, what are we covering? So what we do is we go to are those kids joining the DB kids joining? Just check. So we go here, new script, right? And new script, and then I just paste what I've copied. So I go here and maybe I can just do Control V and everything will be pasted here, right? So that whole code which I picked up from this Word file, what I did was I selected from line number 128, and I selected everything which I have up till this line number 231. So almost 100 lines. So these lines not only have the codes, but comments also. So these are like, I guess, 70, 80 lines of codes. And these codes are really simple. We can just go and execute them. So you can put the cursor here, for instance, the cursor is blinking. So what you can do is Control R. So when you do Control R, this code is then executed down there in the R console. So remember, we took the session on R and we talked about this thing is called as console. This thing is your editor where you write your code. So you can write your code. You can even save that code. So you can do file, save as, and anything which you, any name which you give, it will save with an extension .r. So we go to that workshop folder and we do RNA-Seq, RNA. Remember this an RNA-Seq experiment. So RNA-Seq, just you write RNA-Seq and then it will save this file as RNA-Seq.r. So that file is right there, RNA-Seq.r. And this is your code, and now your code has been transported into that editor.
आपकी से हमें R पर आ रही है तो मेरा ख्याल है आपने से word को किया है.
No.
दीदी आपने entire screen share नहीं की.
Entire की है.
आपने सिर्फ word.
So I go back and share the screen again. And now I can go to window, entire screen. Yes, entire screen, and I'm on screen number 2. My apologies for that. So should I repeat?
Yes.
Yes, I should. So what I do is, this is my R console. Now you can see that. So this is our console, and this was the word code. So I copied that word code from line number. Before this line 128, the initial lines have just practice in R. So those people who are new, they can go through that. And this code is well documented. So we have the comments explaining each line. The beautiful part about R is that code goes line by line. My apologies. And this code you can understand yourself. So for example, it says that if you write rep 1 comma 3 will print 1 3 times. If you write rep and then you put a small letter C and then A and B, so A and B are concatenated together, that is concatenation or combine, and then 3 times, so it will write A and B 3 times alternatively. It will write A, B, A, B, A, B. What if you write A 3 times and then B 3 times? So for that you can do rep C, A, B, comma, each equal to 3, right? So you can practice these things. For example, you can copy that thing and you can go to R and you can just paste it here. One thing is you do copy paste. So this way it will print. So basically everything which I'm using down there in this code, how these things work, the functions which are utilized in these codes are actually explained here. And she says we practiced that before. But if not, it's really easy. You can go and practice these things yourself, and R will teach you about those basics, right? And then the actual fun starts from here. So that line number 128 onwards. So that is the code for differential gene expression, right? So how can we get about the differential gene expression, the DGEs, right? So then we go to R, right? And we do file. And then what do we do next is new script. So that editor opens up and then you just do control V. So everything will be pasted here. And then you can save this thing as save as you go to your respective folder. So that folder was on workshop and its name I just put as RNA-Seq and it will save automatically this thing as RNA-Seq.R, right? So that's how it goes on. Okay. So now my code is there, my script is ready, and then I can just simply go line by line and execute these things. In order to execute a code from this editor, you just select a part of the code and that part will be run. Or what you can do is just put cursor in any line, and you just press two keys. Number one, control, and number two, R. Control and R for R. So control R will execute this code down there. And what happens over here that the cursor then moves on to the next line. So it says you call package HR. So HR does this differential gene expression. So we do library HR and then this function will be executed here. And library HR will be loaded in this R session. Normally R comes without loading, right? So loading is where you put something extra. You have these kids, they eat fries and nowadays they have loaded fries. So loaded fries में क्या load करते हैं वो? छोटे बच्चे आ गए हैं यहाँ पे?
Yes, sir.
दानियाल, हाँ, loaded fries क्यों क्या होता है? कोई और चीज उसमें आप load करवाते हैं and then it costs you more. Obviously that box becomes heavy. So similarly R, when it gets these packages, it's loaded and it becomes heavy. Sometimes you want to load something which is not available, like you want him to give you the pizza of some pizza which is not there. So he if he's nice, he will ask you to sit there and then arrange it for you, right? So that thing is, if something is already not here, it will give us an error that library edge are not found. If it does so, you just run this code, BIOC manager, and then install HR. So everything is here in this these lines of code. So people who are working with the differential gene expression or RNA-seq have to go through this for your FYPs. So next what we do is we create a variable or we create or call them as objects. So we create a variable or object named at we name it as raw counts. And on the right side, we are calling a function in R that's called as read.csv. Its name indicates that it will read a CSV file. It will read a CSV file. Which CSV file? Remember, we stored a file yesterday. So that was your CSV file. This one RNA-Seq.csv, and R is good in reading getting CSV. So data analysis Excel sheets, I guess most of the tools, they love them to be in this CSV format, CSV format. Okay. What was CSV format in CSV? And then this Windows is smart, so it also open up that CSV in this this side view, and it tells us this file looks like this, right? So when you go to, for example, some Excel file, so here you can see those boxes. Actually, in reality, those files, yeah, so Excel file looks like this, and CSV is quite a black and white format, right? And now he's opening up the real file. If you double click, since you have already installed Microsoft Excel, so this file will open up in Microsoft Excel, right? Directly. You won't be able to see what we have in this file. So this is opening on my other screen. So let me bring it here. So it looks like this. So you did not know what exactly CSV is, but if you just click here and here, it will teach you that this file looks like this. So actually the actual structure of file is where we have the separators as these commas, right? So you can take a CSV file and you can save it again as txt. So when you save it, for example, you go like file and then you go like save as, and then you go like workshop, and then what you do is tab delimited txt, and then when you save it, it will be saved like this. So this file is where is it? This one. So that is it's not displayed here. So we double click and it opens up with a text editor. So here we see that we have tab characters here between different things. So separator over here is tab in comma separator, and then end of line character is here that indicates now you go to the next line. That means this is the first one, so that goes here. This is the second one, it goes here. Third one, it goes here. So obviously these separators, these new line characters are important. These file types are important for any tool to get those files in. So one of the favorites of R is CSV. When we go to Python, for Python, you need to first import another library called as pandas, and then the function is pandas underscore, what was that? Read pandas dot read underscore CSV, right? So that is something like my apologies. But R is really good in reading these files. So it directly, the function is, you don't need to import any panda shanda. So it's like read.csv, and then you can just put it here. For example, I can just put up, I can write it here. So I can do read.csv, right? CSV, and then I put those parentheses, and within double quotes, I write just R, and then I press tab. So it gives me the mashwara, right? So I think it's getting slow. So I put another R and then N, right? So RNA-Seq, the file name is RNA-Seq dot CSV. So I can type it, but I can use the script which is already there, which somebody, sorry, CSV. That is the issue with the typing, right? So we hate up typing the things ourselves. So it says, what was the name of the file and where I stored? Oh, one mistake I'm doing. He says there is no such file or directory. What error is this? Can anybody tell me right now? When I started R, I did not do one thing. What was that? Those people who have taken our Linux things, they can quickly point out to what I'm doing wrong because I'm not getting access to my required file.
सर इसमें path नहीं दिया हुआ उस
Yeah, so I did not set the path. I did not go to the right place where my file is. So where I am, the command in Linux was pwd, right? Print working directory. Here we write get working directory, get wd. But since R takes each thing as a function, so you need to put those parentheses, the open and closing. But he says you are sitting in C users, Dr. Wasim and documents. Where is my file? My file is there. So I can go directly, but I can go with another function. Let's go with that function. So it says set wd. And you can ask R the path of the file. So R can tell you where the file is. We know that it's on desktop, but then we need to have the full path. So I can simply click on the file and I do right click and then I do properties, and then here is my path. So C users, Dr. Wasim, desktop workshop, right? And I just copy and then I go back and I can put it here. Or I can put it here as well. Before that RNA-Seq and I put everything here. So what I can do is quotes and I do Control V, but I need to do one thing. What is that? When I copy from path, there is one thing that I need to change. What is that? Anybody? My cursor is pointing you that thing, right? So here the slashes are forward and in the copy paste thing, the slashes are reverse. आप ये डंडे सीधे कर दें, right? So yeah, you should, it's forward slashes. These are not opposite to that. So what we can do is then I do enter. If this function is executed, again, I do up arrow key. The previous command comes in and it tells you that now your directory is set. So that was the hardest way. Easiest way is like you can go to file and there is just change directory. In our times while we were doing, that was my first encounter with R. So at that time, these things were not that easy. But now there is one thing that you can do that. So we used to go with this way. So now my directory is set, so I can recall the functions with up arrow key, read.csv, rnsec.csv. So what happened over here is R just printed that whole file and it ran away. I don't want him to run away. So what I did over here is I store this in a variable. I call it as raw counts. R call these things as these variables as not variables, but it calls them as objects. So this object we create, the name is our accounts. And then we can see what type of object is this. So again, data types, they will come back. So we do is, so that part is where we take that thing. Header.true, we used to do that before because the first line in good old days, so I still have the habit in writing the codes, but just to tell you that previously this header line, this first line of the file, we know it's a header. So, but we need to use to tell R that header is equal to true. So the first line is header. Sometimes this first line is missed, so it's not there. So data exactly starts from second line. So over there you need to do header dot header is equal to false. By default, you can check the default settings of R. So you can go like read question mark and you write that function read.csv. So that will tell you the default settings. So it comes to my other screen, so I can bring it. So that function, this question mark is for help. So basically read.table was actually function. So amongst them, there was this collection. One of them is read.csv. So read.csv goes like this, and these are the default settings. Header is always true. By default, separator is used normally is comma. And yeah, quotations or quotes are with this sign, that thing, and so on, right? So that's how these are the default settings. So here, and again, one thing which we did over here was, let me go without it once to tell you that what we are doing. And we just go like this. You can select on right side of the code and do Control R. So only that piece of code will be executed. If you don't select anything, that means the whole line will be executed. So now he's not printing, but he's storing it in an object named as raw counts. We ask R how many objects so far I have created. So the command is ls. So next line, the command is ls. So I do Control R. It says you have created only one object. That object name is raw counts, right? And then we do class over raw counts. So that is, it says data frame. Data frame is a what type of object? So normally, I think we covered that in the notes. That thing is there. So if you have only numbers, your file is matrix. But if you have in a file numbers as well as the characters, like in this file, we have those gene IDs, they are text characters here, but numbers are also here. So that data structure that stores this thing in R is they call it as data frame. So when R reads the files, looking into the data it contains, it assigns them a data type. So that data type it assigned or that we can get from this function class, it tells us that this is basically a data frame object. Okay, so demo raw counts. Some of the functions they operate only on matrices. So here down there, the programmers they can understand. Rest of them they can just implement this thing as such. So raw counts we took it this file and then we took it as dot matrix. So that converts that raw count file into a matrix file. So that function heat map only runs if it is in matrix format. So we'll cover that when we go there. So class ho gaya, then dim will tell us the dimensionality. So 5792 rows, 7 columns. Head will tell us the header line, the top few, 5 to 6 lines, 6 lines. So head will show the head top part. Raw counts 1, 5. If you specify, like maybe we want to go and see top 15 lines, so we can do 1, that thing is where we chop a file. So that is slice a file, take a subset of a file. So raw counts, and then we break the file from 1 to 15. So that will print 1 to 15 rows. So anything after comma, it will print the columns. So before comma, rows and commas. After comma, columns. In Excel, we go with the columns first. So it says A3, A4, right? So columns and then the row name to get the index position of some data point. So we do here. So we go for 15 lines. Raw counts 1 by 5, 5 lines and 3 columns. So control R, 5 lines, 3 columns. So as you can see, there is one gene ID column that's repeating here. So that is actually included in your data set. So what happens is normally when we apply different functions all the time, we have to exclude this thing. Remember yesterday we took a small primer on R, so where I was excluding this first column, right? So how can we get rid of that thing permanently? Another thing is the rows are numbered, right? 1 and 2 and 3 and 4 and 5. Later on, when we do this DG thing, differential gene expression, and then we come up the list of genes which are differentially expressed, R will report only the row labels. We call them as row labels, right? So 1, 2, 3, 4, 5, 6. So we can set row labels on first column. So I go back to my original function, right? So here it was row dot names equal to 1. So now I run it again. So what will happen? I run it again. I go like ls, I go like class, data frame dim. Now 5792. And here we got how many columns? 7. So before that we had 5792 versus 7. Here we got 5792 versus and 6 columns, 5792 rows. These gene IDs are records, are tuples, the database people, right? And then 5792 rows and 6 columns. Okay. So where that one column gone, that column is fixed as your row labels. So now what we do that, so here it's not 1, 2, and 3, but these gene IDs are set as your row labels, right? So labels are now fixed on those whatever we had in column number 1. For row 1 in column number 1, it was 51746. So that thing is fixed here. This is 9400's row. Later on, the R will return these gene IDs while we do this differential gene expression. So we don't need to then, if it was to sending us those numbers, again, we need to look back to the original file. So what gene existed at row number 2, what was there at row number 5. So in order to get rid of that, right? So again, we do raw counts and then 1 to 5 rows, 1 to 5 rows and 1 to 3 columns. That means wild type 1, 2, and 3. Tail can show you the bottom few lines. If you want to just pull up one column, so you can go like raw counts dollar. So when you write dollar, it starts suggesting. So for example, what I can do is raw counts, and then I do dollar sign and then press tab. So it start giving me advices. So with dollar, you can pull up WT1, WT2, WT3. So here I was pulling, for example, WT3 or 1 or anything. So it will pull up that column for me. Okay. All right. So, okay. So what we do next is we go here. Summary or raw counts will give us the summaries. So that is the summaries for different samples. I can get more. So I'll type 1 and 2, and let me make it a little bit wider and let me recall that function again. So summary or raw counts, right? So while type are here. So minimum is 0. First quartile 8, 41, 1646, 253, 1.3 million is the maximum count, right? These uh, tables, they are better than text, but figures are way better than them. So let's take the box plots for instance. So we go box plot over raw count. So that will make their box plot. So here we get that box plot, but what happened over here? The boxes are not visible. So what happened to my data? We covered that yesterday. So those of you who have taken this class yesterday, they can tell me what happened, and I can take a couple of sips of water. So just think over it, why this box plot is not showing us the boxes, right? And they seem like quite weird, why it is so. Nobody? How many of you have taken yesterday's class? I covered that part. So that box plot looks quite weird. Why it is so? Well, type 1, 2. What do you see on x-axis? Is there sample IDs, names? On y-axis, the range of numbers. Since we have very big numbers in the data set, something like 1.6, what is that? I guess 17 or 8. Yeah, 1. How would you read that? 1.5 is this, and then this next one can be 1.7 million. So that number is quite big. So there was one data point that was getting counts as 1.7 million. Somebody was getting 1.6 million. Somebody was getting 1.5 million. So we have the millions here, and we have the numbers like 1, 2, 3, 4, 5, 6, right? The first quartile is just 8. So 25% of the population is right here. And again, median is 41. That is mean 50% of the population is right here. Third quartile, 75% of the population is also lying here. So they're not that visible because this graph has a scale that is quite weird. It goes from 0 to 1.7 million. So what choice we can have here? We covered that yesterday. We can transform this data into log to the base 2 scale. So for that, in R, you just put a small additional thing. You put another bracket here, parenthesis here, right? So first it will do this log2 or raw counts. We can select that part and do control R. So that part will be executed. Negative infinity is for all those numbers which are zeros. So now these raw numbers are converted into powers of 2. Okay, negative infinity is for 0. So negative infinity का log 2 लेंगे तो that will become sorry, 0 का log 2 लेंगे तो it will become negative infinity, right? So now this part will do the log transformation on data, and then we call a function box plot on it. So again, nested. So nested, right? So parenthesis within parenthesis. This thing was nested programming. I think we covered that. So that the code is written within parentheses, brackets within brackets, right? So first, this thing will be executed. Okay, so this raw counts, they will be converted into the log2. In log2 scale, they will be transformed, and that box plot will be called on that transformed data, right? So we do like this. So what we do is control R. So everything is here, and he gives us some warning, and we give it a damn. I'm not interested into that. Negative infinity is an outlier, so I'm not plotting that. I'm okay with that. But good old days, while I was doing it during my PhD, we needed to add a small constant here. So that is, otherwise it was not printing, right? So that, for example, it was like this, comma, sorry, raw counts plus 0.0 1 or 0.001. So any small fraction 1, so that zeros they become non-zeros, right? And then you apply that function, that warning will be gone, right? But obviously your data then starts from somewhere here, right? And these things are additional here. So earlier it was not working, but now this thing is working. So what we do is we ignore that. So we don't add that 0 point this thing. And then here what we are doing is I'm adding different things or different options in it, right? Different flavors in it. So within those fries, I'm adding ketchup and what's that? The mayonnaise and whatever, right? So we are adding colors to this thing. So again, and then we are labeling the x-axis and y-axis here. X and y are not labeled. We can label them, and you can change as per your own wish. If you have your own data and these names are different here, you can change here. You can change the y labels. You can change the x labels. But this, oh, we did that. So when we execute, goes like this. Warnings are there, but this graph looks lovely. 0 is log to the base 2 में 0 is equal to 2 power 1. Okay. If we ignore zeros in this plot, zeros are zeros, but actual raw counts, they start from 1. Either one read is mapping to some gene or some transcript, or it's not mapping. It's not, if nobody's mapping, these raw, so the counts were 0. So if we ignore zeros, are actually the graph starts from 1. So the minimum over here are lower fence basically in that box plot is at 1. So 1 is 2 power 0. 2 power 0 in log scale is 1. 5 is not 5, but it is 2 power 5. So that's 32 plus little bit. I think it was 38 or something. That was 2 power 6 probably, or 5.5. And then you can convert it back and so on. We have their medians here, right? The actual data was here in that function. I can recall that function because I did that log transformation thing, so it went up. So yeah, right. So the numbers and the pictures, right? And then you can go to that picture and you can do it like save as, you know, because that postscript or meta file or bitmap, and you can save it, right? So you can save it as you know, for PDF. There is one code down there, but earlier I think in the previous versions of R, it was save as PDF. So we do save as, yeah, it's there. So save as PDFs, you can do that, right? And when you do that, the advantage of having, like, for example, I call this thing as box plot, and I can just do that. And I can go back, and this my file is right here. Where is box? This one. Okay. And then what I can do is I can zoom in. You see that is a really good quality graph, right? It opens up on my other screen. All right. So in Excel, you export any graphics. What happens when you zoom in? What happens? Oh जी pixel फट गए हैं. तो यहाँ पे that thing is not there. The picture is of really good quality, straight away publication quality. So when you tell them that you generated these graphs in R, accepted. Okay, pictures की बात करो. That paper should have proper everything. Nowadays they are quite hard on publishing. Okay, so you have to do lot many things. Python, I believe, I think that their graphics is not, I mean, I did not like it. Maybe I'm an R guy. So the people in Python can comment on that. Dr. Nazia, are you here? I think R's graphics is much better. Agreed or
Assalamu
not?
alaikum.
क्या हाल है, Dr. Nazia? Are you there? So what are your thoughts? Is not this
Walaikum assalam.
graphics better?
जी बिल्कुल सर, I am of the point of view कि R के graphics are really better भी, plus ये कि this is quite easy to get this very fine graphics. Otherwise for
Yeah,
Python
yeah,
I have to adjust so many things. I can get this quality, but I
And
will have
another
to
thing
work
is
a lot.
you need to borrow everything. Like Python started eating all other programming languages. In our good old days, we had Java and so many. We started with, I think, C++, and we had so many things. And finally, Python came out, and it started doing everything, like serving the people. And actually, I think most of the community that worked in that in Python was the community working in linguistics, languages, language processing, then accounts people, then engineering people, and now everybody is dependent on that. But what happens is then it started borrowing the things from other languages, like import this, import that, and then all the day you are importing like 10 or 20 libraries. And to do this, only simple this boxplot thing, right? Even to read the file, that CSV thing, you have to import pandas as PD, numpy as NP, and something like that, right?
Yes,
So
exactly.
that is one thing. So there are like 4 or 5 lines of code which you need to write to get that thing. In our boxplot, that's it.
Yes.
Okay. So here, can somebody comment on these box plots? The biologists, it's for you, it's your data. So what these boxes they tell you? These were your gene expression data. Okay. So we need to talk about biology behind that. Is gene expression something that is distributed normally? Anybody? स्टैट्स में आके बिल्कुल आप लोग सुन हो जाते हैं. So I'm sorry, I'm not covering stats here, but in the second module, the class next week is all about data analysis and stats. So feel free to join that. You see, what are these things? What are these things? Outliers. They were dominating these distributions, right? So you have to log transform that, give them the space, and give that poor chaps that almost 100% of the population or 99% of the population is lying here within the boundary of these box plots, within those fences, that 1% at the top is dominating. And because of that, these distributions are highly skewed, right? How can you know that a population is skewed to the right or skewed to the left? What we do next is, maybe I can go further. Achha, first comment is, let me bring the means here. So next line puts the means also like diamonds on these box plots. So here is your median, here is your mean, median, mean for everybody. And I told you yesterday that mean is mean, right? What does that mean? The overall distribution tells us that 50% of the population has the counts like 38, 39, 40 or something around 38 to 40, right? But means were something like 1600, 1700, 1800, and so on. Why? Because we have those big data sets in this number. So that means this distribution is highly skewed to the right or skewed to the left. If you guys have covered the stats part, skewness that we cover in first term, so the distribution is skewed, right? These are not perfect bell-shaped curves. Okay. So what we look for here is if means are here on top of the medians, it's right skewed. If means are here beneath those medians, then the distribution is left skewed. Skewed means more data on the left tail. Left tail means the small numbers. Right tail mean the large numbers. Let me show you that thing as well. Okay, so 3 of them, the wild type one, the control group seems like expression is good, okay, not normal, but close to each other. So these 3 data points are close, like these 3 distributions are closed. Here, the treatment group is this guy get less medians, this guy, we compare medians normally in those box plots, slightly higher, and this goes down. That always happen in an experimental group. When you're applying some treatment, that treatment is good, his hand was good, here again good, here maybe he was harsh, or that plant has a bad day, or that animal had a bad day. So overall expression was low in mu3, right? So the control group is consistent, right? But obviously treatment does something, okay? And it seems like for most of them, if we combine these 3 replicates, the overall expression is downregulated, decreased, right? That's what we can see from here, right? But we cannot conclude it unless we see the result of those differential gene expression thing. So now we are doing this data visualization. We are looking into this data in columns. The distributions are in columns for now, but for differential gene expression, we will go row wise. Okay. The next part is, again, we can see densities, the curves. We can draw the curves, right? And we can get the pictures like this. The curves are like this. So these curves are here are mainly the most of those data points. The highest point is at 2 power 5, 32 or something for most of them. This curve was more like the higher expression was higher in this guy. Who's this guy? This is mu2. And this guy is black one is mu1. And this third red one is mu3. The wild type one and wild type 2 and wild type 3 are a little bit shifted backwards. If you see their right side, they are skewed. A long tail on the right. Is it a perfect normal distribution? Bell curve? No. Skewed, highly skewed. And this line continues with the axis, right? So most of them, they are indicating the right skewness. The graph is pointing towards right. Your right, not graph's right. Okay. Again, I repeat, you are right, not graph's right. It's not like your right ventricle and right ventricle and, yeah. So whatever you think right ventricle was actually in the book. So that book is facing to you. So we were like, whatever you are thinking is right is actually on the left. Here, we don't do that discussion. Anything you see on the right is right. So right tail. This curve is right tail. Okay. It's not like that curve is looking to you and you are then deciding. So the curve is pointed to the right, you're right. So it's right tailed. All distributions are skewed to the right. So as we can see from here as well. And then comparing those curves, it's a little bit hard. Comparing those box plots was easier. So that's why those box plots are there. And most of data analysts like me, including myself, we love box plots. Okay, so we reach here. Library dot sizes. What we do is we sum whatever we have on those columns. There's a function called as call sums. So call sums, what it does, it sums whatever we have in the column. So we are summing whatever we have in the column. So we are interested to see how much data was here, 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 here. So this guy seems like a bad day. So 3.8 million here. It's, I guess, what was that? We can convert it into millions. What we do is we just store it into that variable, but just let me tell you in R, how can you convert the thing in millions? So we do up arrow key, you write this function call sums or raw counts divided by 1 ke aage 1, 2, 3, 4, 5, 6 zeros. So this is 9.5 million, this is 11 million, 10 million, 10, 11, 3.82, right? So that guy has a bad day. So overall counts are low. So here what we are doing, we are taking those files, like for example, we are taking all those numbers. And let me pull up the head part, head over raw counts. Okay. Oh, my ball. Oh, sorry. Spellings are screwed up. So counts. Right. So these numbers 436, 4929, everything what we have in this column, and we have 6,000, how many? 5792, around 6,000 data points here and here and here. They are all added up into these numbers. So that number, this column, sum of everything in this column is 9.534 whatever millions, right? So that means the expression is overall low. So we store that information into a variable. We call it as library.sizes. And this will come again in when we do that differential gene expression thing. Another thing is bar plot. We can see those call sums in bar plots rather than numbers. We can go into the bar plot, and this looks like this. So again, wild type 1. Here the scale tells us that this thing is around around 1 million. Actually, it was 9 point something million. That was that was around, I think, 11 point something. This was again around 11, 12, something like that was that 3.8 something. So we are seeing these things as a shape of a bar chart. Okay. I'm sorry. I don't want to close that thing. Okay, so here we are. So bar plot heat map, one of my favorites. So heat map. Actually, what it does is, let me make it smaller. I can keep it here. Yeah, रख लेते हैं इसको, and then I can here. This is the beauty of this standalone R in R Studio. These panels are fixed, so you need to can do like this all the time. Okay, here you just grab it and then you can move it freely, and you can put these things on top of each other. So this thing is, so what heat map is doing, heat map is showing these numbers into a what we call it as a heat diagram. So low heat, white, and then red is where there is more heat. Initially, when it started, it was other way around. Red was low heat, white was more heat. The reason behind that was the sun is hottest at 12 p.m. So at that time, its color is white. During the early mornings and during the time of the sunset, that sun color was red. But now they fixed that actually. So the low numbers and high numbers, they are here. So for example, this guy 81G01240 in wild type 3 has high expression in all three, wild type 1, 2, and 3. And in mutants, the expression is low. Obviously, this thing is then down regulated in the mutants, up in the wild types. Okay. The top there is this cluster diagram. Here he's saying that your data is split into two clusters, mu ones and wild type ones. They are separated. So that means your treatment is making a difference. So there is these wild type ki jo all three replicates, they are close to one another, and mutant ke replicates are close to one another. So your data is clearly separated into two groups. That means this treatment is making a difference. There is some difference of gene expression. We will go and do that DG thing and see how many are differentially expressed. Okay. All right. So what if I don't go for only, by the way, so I took heat map for top 10 genes. So what I can do is I can remove that part and I can get the whole pattern. It will take some time, but yeah, let's give him some time. So he's replacing this picture. He's running, so that picture will be changed as soon as he's done. Up till now, if you have any questions or queries, you may ask. We have 1 hour, and I think I will be able to end this DGE part in a moment. So here, all those genes, again, the genes which are close in expression to each other are together. So 1300 and 8870, their expression is close to one another, and so on. If I have your, for example, class results, and I take that. I don't know you, most of you, the kids in my class databases, right? And then if I make your heat diagram, so your friends, the people who are, in groups, right? They will be clustered close to each other. Acha, top there is, for example, these are your subjects. Again, here, these are those samples, mu1 and mu2 and mu3. Here we see very interesting that this mu3 is entirely separated from rest of the experiment. Wild type, the replicates are close. Mu1 and mu2 are close, but mu3 is separate. So sometimes what we do is we then discard that sample who is not at its right place. But here again, we have only 3 samples, so we will retain them. In my real experiment, I have like 200 samples. So at one place, we actually discarded, I guess, 27 to 28 samples because of that thing. Okay, because you are applying a treatment, but there is some possibility of variation within the experiment. So what happened was the graduate student, while he was sampling those plants, he gave those samples to an undergrad student, and she got late in the lab. As a result, their gene expression changed, right? So you take those samples out of the refrigerator, because in RNA-seq experiments, we preserve RNA wherever it is, right? So in animals, we used to put in, I guess, at that time it was formalin. And for plants, we use liquid nitrogen. Now, you can put in liquid nitrogen, the animal samples as well, right? And then those things are preserved, right? So your gene expression RNA is preserved. Otherwise, it changes real quick. Fractions of even, I don't know, maybe nanoseconds. Okay, so that is where you need to while you're sampling, you need to stop that gene expression at some point. Gee, Nazia, you have a comment? Dr. Nazia
जी सर
from
मेरा एक
SEEKS
क्वेश्चन था.
School of Electrical Engineering.
जी सर सर
जी
एक्चुअली
डॉक्टर नादिया, बल्कि आप भी अपना कोई ये मॉड्यूल बनाए जो हमारा सेकंड वाला है ना, उसमें आप क्या कंट्रीब्यूट कर सकते हैं। आपको टॉपिक्स तो आपको दिखाए हुए हैं वहाँ पे।
बिल्कुल ठीक हो गया सर, इंशाल्लाह उस पर आई विल वर्क ऑन इट सर। ये वाला एक्सक्यूज मी, मे बी
जी जी
मैंने
प्लीज।
बिल्कुल स्टार्ट से ज्वाइन नहीं किया, तो आई एम गेटिंग ऑल द पॉइंट्स जो अभी हम कंप्यूटेशंस कर रहे हैं, जो हम कैलकुलेशंस वट एवर वी आर डिस्प्लेइंग, बट आई एम मिसिंग द बिगर पिक्चर कि इसमें जो ये वाइल्ड टाइप जीन्स हैं, दीज एंड म्यूटेशंस आर, मतलब हम बेसिकली व्हाट आर वी गेटिंग रिस्पांस टू सम ड्रग, या फिर we are the what is the bigger picture, हम ये को वाइल्ड टाइप
Any,
जीन्स को
it
क्या
can be
करते हैं?
any experiment, any experiment. So this experiment has two groups. So we have the wild types and we have the mutants. Since I can go to the original file and just briefly explain what happens is these are the gene expression values, right? So what we did was we set up an experiment where we had six plants, or maybe somebody has animals, so things are similar. So and why we do that in replicates, why we did on mutants, we did three readings or three samples, because in order to do statistics we need replicates, right? So that part we also covered yesterday. If it's only one sample, you cannot do you cannot calculate the standard deviation, right? Mean is average, you can calculate the means, but you cannot calculate the standard deviation. So what happened was we took those six plants in two groups. One was assigned no treatment. Second group was given some treatment, some radiations or some chemicals or some reagents. And then we extracted their RNA, and then we sequenced them with the help of NGS technology. And then after this sequencing, what we do is we took those RNA fragments from those sequencers and map them or align them on their reference genes or transcripts. So this guy got 436 and wild type 1. Same guy, this gene got 536 here, 776 here, 4 here, 10 here, 3 here. That means in the mutant group, it was down regulated. Its expression is gone down. This is the gene expression values. The DNA makes RNA. We call this thing as gene expression. We makes RNAs first, and then RNAs are then translated into proteins, right? So that complete gene expression is up till protein level, but this thing is we are going up till RNA level only. Okay, so that was our experiment. So when we got this
Okay, thank you.
this heat map, we observed that the mutant groups are much variable from each other. Two of their replicates or two of their samples are close to one another, like this here. And then even wild type 2, as per this default settings of this heat map, you can go and check the function heat map, what it's doing. I think he's going with Pearson correlations, point-to-point correlations of these. So I have 6 data points here, these 6 samples, and he's correlating which one is correlated to which one. Okay, how strong they are correlated, what are their correlations, and then he's placing them on the basis of closeness to each other, clustering, right? And this guy mu3 seems like different from rest of them. So this is what we are doing right now. This overall data separation, we can call it maybe, or visualization, right? So how our samples are behaving. Okay, now we are going to the second part. Again, here there are different variants of this heat map function. In one, we remove that, okay, we don't want this clustering on row wise. We don't want this clustering on columns, so different results we can get. Let me put this thing here. So here we don't want clustering either on rows and columns. We just want to print data as such, and we just want to see its picture as such. Okay. All right, so library HR. So we reach up till HR. So where we do this differential gene expression. So for that, HR needs three things: counts, the raw counts, matrix of counts, that is raw counts. It also needs the experiment. We pass on to a variable. We call it as group. So the information about our experiment, we know that this is our experiment was set up. So we had wild types and mutants, but R does not know. So we need to teach R this whole experimental design. So for that, we create something like here. So we do rep C over mu1 and wild type. You can do the anatomy of this code. You can just, take its different sections like copy and you select up till here, C mu1 comma wild 2, sorry, WT. So that will make, we do control R. So it will make a list of two things, right? Mu1 and wild type, right? Find them together, concatenate them together. So that function small letter C is combine or concatenate. So we can ask R what this function is about. You can do like this, right? And you can, it says combine. It's a generic function which combines its argument C, right? C can be combine or it can be concatenate, right? So C is a function that is combined. So combine these two things together. Rep means replicate this thing and each equal to 3. So this part, if you select up till here, right? So it goes mu1, 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 wild type, wild type, wild type. So your experiment looks like, right? Wild type, wild type, wild type, mu1, mu2, and mu3. But we know that mu1, 2, and 3 belongs to mu1, and wild type 1, 2, and 3 belongs to wild type. Okay. What we do next is you can store it in a variable. We call it as expt. Expt means experiment. So we do control R, right? We do again control R, and he will print that thing again for you. There is one thing here called as factor. So here we know that there are two groups, mu1 and wild type. These three belong to mu1. These three belong to wild type. Uh, have you guys covered the factors? What are factors in stats? In stats, everything is called as variables. If gender is variable, how many possibilities are there? There are two possibilities, or if you include transgenders, there are three possibilities. So these three things are called as factors, right? Or levels, sorry, levels of that factor. So only one factor is here, but there are three levels, male and female and transgenders, right? Or just male and female. In that case, this is a binomial variable that has two things. So here, when we apply this factor thing, it will factorize these things and give us the levels. So that factor, this list, this expt variable, when you factor that, it will go like this. So we do control R, and it says these three are mu1 and these three are wild type. Okay, mu1 and wild type. Library.sizes is storing the information of the library sizes, the call sums, how much total data we have. So we need these three things. Number one, counts. Number two, group, a factor with length group or expt. We created a variable expt, right? That tells us how many groups of the treatment are there or how many treatment groups are there. And library.sizes is something that stores the information about their call, some how much data we have in each column, the total RNA we have. Acha, by the way, this is RNA, but in another experiment and in another example, this can be something like your different branches of different companies in one city, like Lahore and Karachi, and people's salaries or people's overall hourly payments, right? They seem like low here. Maybe it's Rawalpindi, my city, and this is Islamabad. So Islamabad is heavy. And somebody who worked on three places here and then three places here. We need to find it. What was his growth? Whether he got more improvement in his salaries or he got the decline. Obviously, inflation rate is higher in our country. So we are sort of, in terms of our salaries, we used to tell our friends in USA, well, we came here, it was, I said, what was that? 1,000 US dollars, right? No, I think it's less than that, but in PKR it's increased. But again, um, obviously you cannot directly compare the salaries or the money, but a lot many other things which you have in your life. But anyways, so we pass on all these information into one function that's called as DGE list. So DGE list is a function in HR. What it does, you can just ask here question mark DGE, all capital. I think after L it's capital. It's technical, a little bit hard to understand, but those folks who are doing DGE things in their masters or graduation, bachelors or PhDs, um, You will learn these things. You will see these things again and again and again, like I did that. So that's why I'm a little bit more confident, but you'll be confident if you work on these things. Okay, my objective was to expose you to these things, give you an exposure to these things. Otherwise, if you are not able to understand, just aankhein band karke keep on running these codes. Only file name you need to put in the beginning, and you need to make the changes here in this experimental design. For example, you have 4 replicates and you have 3 or maybe 4 or 5 different levels of the treatment. So in that case, you can change it here. Okay. All right. So we just apply this thing to DG list function, everything. Here it says counts equal to as dot matrix counts. Counts को as a matrix लेना है, not as a data frame. Originally, that object was data frame class over raw counts. The data type is data frame. Okay. And I covered that in heat map also. So this is data frame, but we need to give him as will convert that data type. It's called as, what was that? Typecasting in programming, as dot matrix over raw counts. That's what you need to do. And then give it to this thing, counts equal to, you need to write it. Lib.size equal to, here we pass on the second thing, the second object or variable library.sizes. And in group equal to may, you provide that information, that experimental design. So group equal to expt. You can call this thing as group or GP and then put GP here. So we do that and we assign it to a variable, call it as D. Okay. Now we ask what type of object is D. So again, ls will tell us that you will create object. How many objects you have created? So I created raw counts and now I've created, we created expt. We also created raw counts, library.sizes, and now we have added one more thing called as D, right? So class over D will tell us that what class it has. So it says it's a DGE list object, right? From Azure package. So basically its data type is, it's a DGE list. That's its data type. Names will store the experiment, the information you pass. So you have the counts and then you have the samples information, right? So if you say D dollar counts, so here in D, the things names over D means in D object, we have 2 things, counts and samples. So counts have the information about the counts. How can you get that? Again, you do D dollar and then you write counts. So those count information comes in here, everything. Here I just chop to top 5 rows. Okay. So that's like this. So counts like this. Samples will give us the information about samples. So samples may it stores their groups. So these 3 are mu and these 3 are wild type. Can you give me any error here? Anybody? Can anybody point me some error? Wild type 1 belongs to mu1 and wild type, right? Wild type 1, 2, and 3 belong to mu1 group. No, it's other way around. So I need to go back and I do one correction. Either what I can do is the easiest part or simplest part is in my CSV file. I think I flipped around. So where is my CSV file? So I can go and read that CSV again, or maybe I can fix that here in the code. I just figured it out. There is one small error here. So I can just flip around. Okay. So where is my CSV file? This one, sorry. So what I can do is cut. That's very important because then your whole result will be changed. My apologies. I can fix it in the code as well, but that will make it a little bit uglier for you since you are already understanding the things. So I don't want to run into more code. Okay, so go back and what I can do is this part. Just in a minute. Here I read it again. Oops. then I need to fix this library.sizes thing. Okay. So that's good to go. Down there, I can go back and yeah. So I can restart with this D thing. Rest of the things are fine. EXPT and everything they are fine. So I do D and I do names and class and counts and yeah. Now it's fine. Now it's fine. Okay. All right. So what we do next is D dot calc norm factor. So here it made three columns. One is group. Group variable has the information that this thing belong to this group. Mu1, mu2, and mu3 belong to mutant group, and wild type 1, 2, and 3 belong to wild type group. And this is their library sizes, the total counts they have. This guy has 10 million, this guy has 11, 3.8, 9.5, 11, 10, and so on. Here he had inserted another column called as norm slot factor, norm dot factors. Normalization. Right now, every count will be counted fully. Every one will be counted as one here in all these samples, but he need to adjust that. Some of the samples are big, some of the samples have more genes expression, right? Some of the companies have heavy budget, some of the companies have low budget, so we need to normalize for these things. So there's a function called as calc norms factor. So that will calculate some numbers and go through different samples and adjust their counts by multiplying with these numbers, which we will get it from here. So every number here, if it is 1, it will be taken as 0.92. Here the numbers will be increased. Here the numbers will be slightly increased, decrease, decrease, little bit increase. Why he's doing that? He's going into each column, all the numbers in each column and looking for their means and standard deviations, and he's doing some calculations. That's his own method of normalization, so we ignore that. Right, so now everything is set. So now we go for another plot. This thing is called as multi-dimensional scaling plot. There is a function called as plot mds.dge list, and we give him D, and then we can give him the name and we can change the colors here and we can assign colors to different groups, right? So that when we simply run it, Control R, it will go like this and tells us that 3 dots are here, wild type 1, 2, and 3, and mutant 1 and mutant 2 are here, mu3 is here. How to interpret this graph? So people, if you are familiar with principal component plots, this thing is called as, it's also from here, we are also entering into the next domain, the AI thing. So that's your unsupervised machine learning. So we applied this thing, this function. So the pictures like PCA or multidimensional scaling plot, or I think in the next we have TSNE plots, all those things are where we are doing this unsupervised machine learning. So we gave the data to the code or to the program, and then it looking into the similarities and differences, it separate the things according to whatever it finds. They are similar, they are close to each other. The ones which are similar here, this guy is different, so placed on the another corner. It's like you give these multiple things to a baby, and then baby recognizes the similarity among those things, and they start separating toys on one side, electronic devices on other sides, right? Um, Glasses and dishes on another side. So that's how it goes on. Okay. So how would you interpret that? So that zero line, if you bring it here, this zero line, you bring it here, you can make it 4 sections, the coordinate things. The things which are close to one another are in fact closed, right? So the gene expression pattern within these 3 are close to one another. These 2 are close. And this guy seems like different. Either if I had more samples like 5 here or 5 here, I would have removed that. Why I'm not removing? Because already I'm lack of replicates. So when I do this statistical testing, if I throw away the replicates, I will lose the power, statistical power, right? Power to reject the null hypothesis, which is wrong. That's not the topic of this discussion, but that is one thing very important. You guys should pay attention to when she comes to this type 1 errors or type 2s or powers. Okay, so what we do is this graph tells us that this mu3 is entirely different from rest of the group. We move on to, I'm not discarding that, but I'm moving on to last part, which is DGE testing. So here it applies another function, estimate common dispersion. There is a common variance or dispersion. Here he's calling dispersion, right? So that all data points, if you take their grand mean and then you calculate a standard deviation from all of them, so that's your common standard deviation or common variance, right? So he's doing something similar for the whole file. All those data points, 5792 multiplied by 6. How many? So that many, let's suppose 6,000 multiplied by 6. So we have 36,000 data points in our experiment. So that's what we are doing. So we are calculating a common dispersion. And now we can go for that test, statistical test. Here the test is NBT, negative binomial test. Negative binomial test. Basically, negative binomial is the type of distribution. The actual test is Fisher exact test by R.A. Fisher. That is where your data is not normal. This data is, they assume, binomially distributed. If you guys have covered the binomial distributions, fine, but otherwise, a situation is binomial if you are having one of the two things. If you're collecting the data about dead or alive, either something will be dead or something will be alive. So it's one of the two things. There's nothing in between. Okay. You are collecting the number of students in a class. So that's a discrete thing. Okay. Gender, it can be either male or female, right? So male or female. So that is one of the two things. Again, the thing is binomial. Okay. So in binomial models, we go for P for probability of success. In negative binomial, we actually use Q, the probability of failure. So that's why these things are called as negative binomial models. RNA-Seq thing is something that follows a binomial model, negative or positive, that's secondary. So binomial in the sense that when you take a read fragment and you align to some gene model, either it will align or it will not align. Okay, so again, there is a binomial thing. Either it will align or it will not align. Okay, so whole read will align. You put +1 to the score for that gene. If not aligned, you don't put anything. Okay. So that is where we are applying a test. Here, this is the call which we are giving. So pair between wild type and mutant. So it will do mutant minus wild type. Mutant as numerator, wild type as denominator. Numerator, denominator. Okay. And then we give it to this D. D is that variable which we have prepared. We have normalized. We have made it ready. Okay, and it is ready to go into here in this function, exact test, no, right? So exact test D pair C this thing, and then we assign it to something as NBT, another variable or object I'm creating because that thing will be used down there. Okay, so we apply that. So test is done. Names over NBT. So NBT is we created an object called as NBT. We ask for its class. So class over NBT tells us that it's again a DGE exact, basically exact test. So differential gene expression के लिए एक you applied an exact test. So that object is coming from library HR, and it again has three things. So we have NBT dollar. What was that? So yeah, names over NBT will tell us what it contains. So names will tell us what it contains. So it has table, comparison, and genes. And we can look into and check what we have in this. So table 1:6 will have this thing, right? So we have the log fold change of all those. So all those 6 data points and their average log fold change. Log CPM is their counts per million. So each data point has the counts. Okay. But those counts are coming from 11 million. Somebody is getting from 4 million. Somebody is getting from 3 million. Different samples have variation. So what R does is, or what HR does is, the accounts for this thing as well. So it converts those counts into per million. So how much you are getting if your country's budget is in millions, right? Some countries' budget is in billions. So obviously, as per their economy, they are doing good or bad, right? You cannot directly compare somebody who's getting the salary in PKR versus Saudi Riyals versus the US dollars, right? So everybody has different matrices to evaluate. Okay. All right. So NBT dot corrected here. When you do that, it will do that multiple test correction. And here we see that we got another column here. So that is this FDR. So now you ignore the p-values, you look here. FDR should be less than 0.05, not the p-values. Why we are doing that? Because this thing we introduced here is called as multiplicity problem, multiple test problem, right? So you are setting up like so many experiments. You are throwing 6,000 tests, 6,000 rows. So those false positive rate or false discovery rate increases. So here we do that FDR correction, right, to minimize the false discovery rate. So that p-value that was 10 power minus 263 now has been little bit raised higher to 10 power minus 260. Obviously, these are fractions and very small numbers, but something like close to 0.05. So if somebody has a p-value of close 0.049 in FDR, it will be raised to 0.051. So that gene that is there on the borderline, you will not pick up. In fact, you want to reduce the false positives. We don't want that. So for that, now they advise that after applying this function, you should look into this column. So any gene less than FTR equal to 5.05 is what we call it as differentially expressed. Okay. So NBT dot table, and then we go with this. Where were we? I think here. So now we pass on that information, this NBT dot corrected dollar table, the whole table to another object. I call it as NBT table, right? And on NBT table, I put a condition that NBT table dot FDR dollar FDR should less than 0.05. So all those rows that have in FDR column p-value less than 0.05 will be selected and pass on to another object DEG, right? And when we had DEG, I see it looks like this. So only differentially expressed genes are here. I can count how many are them. They are there. So we go with either head or we go with dim. So it says 921 are differentially expressed, right? And we have these 4 columns and 921 rows. So 921 are differentially expressed. How many were differentially expressed yesterday while we were doing t-test? Now we did a proper test and we got more. How many we were getting while we're doing t-test? Yesterday, when we did this analysis in Microsoft Excel, how many genes finally we retained? Can anybody remember that? How many we retained yesterday? I guess they were like 320 something, 321 or something. Okay, now we got 3 times more than that because we applied the proper test. So for RNA-seq data, Excel is not good. T-test is rejected because it's not a normal distribution. The assumption for t-test is that data is following a normal distribution, where here the data is not normal. Okay. All right. So we can check how many are differentially expressed at this cutoff. So sum over this thing will tell us that total 921. DEG $FDR less than 0.05. When you do that, it will give you the take all those genes and pick up only that are meeting that condition, true ones, and their sum of all those true cases will return 921 in this function. Okay. All right. So you can report these things into a file. You can do write.csv. While you do read CSV, you can also write this whole result into a file. We call it as CSV, deg.csv, control R, right? And then we can go and see we have created an Excel file. We named it as what was that? This one, DEG. Right. So we just open. So these are the genes which are differentially expressed. Okay? So that's differentially expressed ones. The ones with the so the ones with the fold chain positive are upregulated and the one with the fold chain negative are downregulated. Okay. So that's all. I think that's where we end up this first part, and then we move on to this ML things. We start with the ML on gene expression data. So I hope maybe the people in the first course they can stay, because I think there was this ML part on RNA-Seq that was also there. All right. So that's it. So this is how we do this differential gene expression.